So the reactions of carbonyl compounds. Now, I think you guys would have already looked at the carbonyl compounds already, right? Have an understanding of what the carbonyl compounds are in a sense, right? And we're gonna be looking at some reactions, and let's look at the general objectives that we have. So on completion of the section, students must be able to describe the structure of aldehydes and ketones as they are carbonyl compounds. We must describe the reaction of we must be able, rather, to describe the reactions of carbonyl compounds with Brady's reagent, right? Tollens reagent and Fehling solutions. We must be able to describe the reaction of carbonyl compounds with acidified potassium permanganate and potassium dichromate. And we must be able to describe the chemical reactions of carbonyl compounds A, reacting with a sodium cyanate in the presence of hydrochloric acid, all right? And B, reduction of the aldehyde to primary alcohols by lithium aluminohydride hydride complexes or um, platinum catalysts, right? And the reduction of ketones to secondary alcohols using the same LiAlH4, right, with platinum catalysts. The last thing you should be able to do is to explain the steps involved in the mechanisms of selected chemical reactions of carbonyl compounds. It's really what's happening there in 4A, right? So the reaction of carbonyl compounds with sodium cyanate, all right? So that's basically nucleophilic addition mechanism. So that's like how many mechanisms we do so far? We did one, two, three, four. I think this is the fifth mechanism we're looking at now. All right. So carbonyl compounds. So these are groups of organic compounds containing carbon atom doubly bonded to the oxygen atom, right? So this is a carbonyl, group. Can anybody tell me what functional group this is though? Which functional group is this? All right, it seems as if we weren't ready yet. So let me re-ask the question. Which functional group is displayed on the screen right now?
Is it a ketone, sir?
Are you asking or are you telling?
Talent?
You're telling me that it's a ketone. It is a ketone. All right, so that's a ketone there. All right, why did you think ketone?
Because of the two R groups.
All right. So if you know it's a ketone, don't ask if it's a ketone. Let us know that it is a ketone. All right. Confidence is key. So here are different types of carbonyl compounds that we may or may not have encountered. Aldehydes and ketones are what is going to be exemplified within this entire PowerPoint as our carbonyl compounds. Aldehydes and in al, ketones and in on. We have carboxylic acids, which are also carbonyl compounds. Acid halides. We're going to be looking at the reactions of acid halides, right? And we're going to also be looking, well, I don't think in CAPE we look at the reactions of acid anhydrides. I don't think so. Acyl phosphates are present as well. We also have esters. We know that, all right? Amides as well, right? We don't really look at lactams or lactones or thioesters, right? But there are examples there. So the ones that we normally assess in CAPE are aldehydes. Interesting. Give me a second. Let me just do this. So the ones that we normally look at in CAPE are aldehydes, are ketones, are carboxylic acids, are acid halides, right? Are esters and are amides. Those are carbonyl compounds for this chemistry, right? So everybody should be familiar with them or at least get familiarized with them by now. All right. So let's have a look at the rest of it now. So carbonyl chemistry is interesting, all right? And we know that in carbonyl compounds they have this double bonded carbon and oxygen group, right? We must be able to realize or to figure out that this group is present, right? With alkanes, we know that if we add um, molecular halides to alkanes under the presence of UV light, it should decolorize the solution because free radical substitution will occur. right? We know with alkenes, it doesn't require the light. If you leave it in a dark room, it should still decolorize, right? So we know that is the presence of the double bond, right? With alcohols, we know the Lucas test and we know Jones test, right? And we know the colors that's supposed to be coming out. So we know how to identify whether or not something is an alcohol. Now we must be able to identify whether or not it is carbonyl in nature, all right? And for that, we use 2,4-dinitrophenylhydrazine, right? So testing for aldehydes and ketones in this case, right? So for carbonyl group in this case, right, we find carbonyl compounds, right, more namely aldehydes and ketones by using 2,4-dinitrophenylhydrazine. All right. So it's an orange and yellow precipitate that will be formed, right, when it's added to an aldehyde or a ketone. So that's how we know whether or not the substance is an aldehyde or a ketone. This is a presumptive test, by the way, right? And our presumptive tests are the tests that we look at in functional group analysis because all of functional group analysis are presumptive tests, right? Our complete and definite tests will be through titrations and obviously through UV spec analysis, right? Mass spec analysis and those type of stuff, right? But no, we're just presuming that if we react to 4-dinitrophenylhydrazine with a compound and it gives us this orange yellowish precipitate, then there might be an aldehyde or a ketone in there, right? How will we know for sure? UV spec, IR spec, mass spec, right? But we're going to speak about that in module 2. So it only happens with the presence of the carbon to the double bonded to oxygen, that type of group, right? And not in more complicated carbonyl compounds such as the carboxylic acids, right? And it's not really that dreadful, right? For it not to be able to really describe these substances. Because we can look at the different types of carboxylic acids and we can define whether or not it's carboxylic in nature, right? We can use different tests. And we're going to be looking at that in the reactions of carboxylic acids. And if it is strong enough, we can always do a litmus test, right? Or use universal indicator solution or phenolphthalein or anything like that, right? So shouldn't be too bad. So we have that precipitate that is derived. The purification of the precipitation by recrystallization can allow us to determine the melting point, and we can identify the particular aldehyde or ketone. So aldehydes and ketones are volatile compounds, right? Meaning that melting point and boiling point are really key physical characteristics of volatile compounds. And depending on the melting point that we'll find out after purification by recrystallization, right? Then we can figure out which specific aldehyde or ketone this is. All right. So that is it there. Any questions? Just drop ones in the chat if we're ready to move on. By the way, 2,4-dinitrophenylhydrazine is described as Brady's reagent. All right. So let me know if we're ready to move on. Just drop a numero uno. Go ahead.
Hi, good night. So
Good night.
I just joined. Sorry for coming late, but I have a question.
Mm-hmm.
So you said it only happens with C-double-O groups, not more complicated ones. Why doesn't it finally precipitate more complicated ones?
Would be steric endurance. All right. Okay. Okay. So steric endurance is probably the most simple way I can explain why it does not happen in that case. Right? Of course, it's more complicated, and when you're looking at mechanisms and stuff like that, you'll figure out that based off of internal interactions and stuff like that within the molecule, right? The precipitate will not be able to be formed because the dinitrophenylhydrazine wouldn't be able to actually attach to the molecule to let it fall out of solution to form a precipitate. But we can just leave it up to stereochemistry, right? It's not something that we dissect in unit 1, unit 2 rather.
All right, sir, got it.
So let's say stereochemistry. Um, could probably look at why, all right, but that would probably be in, you can just ask me and I'll see if I can explain it to you outside, all right? So let's have a look at this now. We now know how to identify whether or not something is aldehyde or a ketone by using 2,4-dinitrophenylhydrazine, right? Bright orange or yellow precipitate. Everybody's on this, that is understood, right? So if we have two specific solutions, solution A and solution B, and we react to 4-dinitrophenylhydrazine with solution A and solution B, and we get that precipitate. We know that they are carbonyl in nature, and we know that they are not esters or they are not carboxylic acids. So they're most likely aldehydes and ketones, right? But which one? Is it A or B? So we must be able to discern now whether or not, right, something is an aldehyde or ketone after already figuring out that it is carbonyl in nature. So let's have a look at this. The first test is the Tollens test, or what we call the silver mirror test, right? So in the laboratory, the Tollens test may be used to distinguish between the aldehyde and ketone. And make a note of this. Tollens reagent is a solution of silver, right? Silver and silver nitrate, right? So we're gonna have that there, right? Keep that noted, right? So it's silver ions, right? And silver but from silver nitrate, right? And ammonia. So what we call this type of solution is ammonified silver. So if you see anything referring to ammonified silver, this is what we call Tollens reagent, right? So it is the presence of the silver ions from the silver nitrate, right? And ammonia. So it must be ammonified, right? So that means that we add ammonia to the solution. Wonderful. So it oxidizes aldehydes, but not ketones. So positive aldehydes, negative for ketones. So the silver ions are reduced to metallic silver, right? So they gain the electrons, right? The oxidation state is now 1, so it goes back to elemental silver, which is now metallic silver. And it forms a layer called the silver mirror on the inside of the container. So if we look at something like this was a reaction, right, done within the lab, right? So we looked at it and we got this type of test here, right? So the silver deposits, right, the reduced silver deposits, right, would line the surface of the test tube, giving that type of silver mirror, right? But note that if it gives a cloudy black precipitate, that is still positive. All right. A lot of persons make the mistake that if it is not a silver mirror, it is not an aldehyde. That's not the case. Right? It depends on whether or not it is aliphatic or aromatic in nature. Right? If it is aliphatic in nature, it's more likely to create a silver mirror. And if it is aromatic rather in nature, then it's more likely to give us the black precipitate. Right? And it can give us this brownish precipitate. But if you don't say anything, then it must be negative. If there's no reaction, then there must be no carbonyl group or no aldehyde functional group present. All right. So we have the reaction here. So an aldehyde reacting with ammonified silver here in the presence of water. This is oxidized, right? So we have the oxidation of the silver to produce this. All right. So we have this ion here, right? And then we have the silver coming out of the solution per se. For ketones now, no reaction, right? Or no reaction should occur. Let me just say should. So that's really it. So after we figure out that both of the compounds are aldehydes or ketones, Tollens test can let us know whether or not it's an aldehyde or a ketone, right? So if it's an aldehyde, it will give a positive reaction with Tollens reagent. If it is a ketone, it will not, because ketones cannot be further oxidized. Well, further oxidized under this condition. Right? My brain had to take a second there whether or not I should say oxidized or reduced, but it's oxidized, right? So we have that there. Any issues? Any concerns? Everything is good for this Tollens test?
Yes, sir.
All right. Wonderful. Moving on then. Fehling's and Benedict solutions, right? Now, I added Benedict solution because it also works with Benedict solution. But we're supposed to be looking at Fehling solution specifically. Now, where else have you seen Fehlings and Benedict solutions being used? You guys have been seeing it from third form. Go ahead.
Just for reducing and non-reducing sugars.
Wonderful. Right? So reducing and non-reducing sugars. So it exemplifies the fact that glucose is most definitely an aldehyde and fructose is a ketone. Right? So there are these functional groups present within our sugars, and that determines whether or not they're easily oxidized, easily reduced or not. Right? So they're whether or not they're reducing sugars or non-reducing sugars. So failing solution and Benedict solutions can be used to figure out whether or not something is an aldehyde or a ketone, right? So remember that failing solution contains copper ions, right, complexed with tartrate ions in sodium hydroxide solution, all right? Benedict solution is copper ions complex with citrate ions in sodium carbonate solution, all right? And the tartrate is acts as a anti-precipitation agent. So it helps the solution remain homogeneous and disallows the precipitation of copper 2 hydroxide, right, in filling solution. And the citrate helps prevent the precipitation or formation of copper 2 carbonate, right? Not going into all of that solution chemistry yet, all right? But generally, no, ketones will give no reaction, no change, right? So if you look at reducing sugars and non-reducing sugars and stuff like that, right? The ones that are that have ketones, right, in their groups, right, it will not actually react. And the ones, right, for example, glucose, which are aldehydes, which actually give this dark red copper 1 oxide precipitate, right? So this blue solution provides this dark red precipitate. And we've seen this before time and time again. All right. And you guys should have explored this a little bit in Unit 1 biology, I believe. Yeah. When it explains the chemistry behind the food tests, right? Module 1 biology, right? So this is how we really look at them, right? So another type of test, the failing solution specifically, right? I added Benedict solution there, as I said, because it also helps, right? So a failing solution, the ketones will not react, the solution will remain blue. Aldehydes will, of course, react, forming a dark red or deep red copper oxide, right? And that is our red precipitate there. All right. So aldehydes are reactive. Here, ketones are not. All right. Any questions? All right. Any questions at all? Okay, moving on. Okay. Oxidation via potassium permanganate. No. Aldehydes, when refluxed with excess acidified potassium permanganate, right? Aldehydes are oxidized to carboxylic acids, right? So when refluxed with potassium permanganate, they're oxidized to carboxylic acids. So we have here now the example of propanol in the presence of an oxidizing agent, which in this case could be potassium permanganate, would give us propanoic acid. All right. Ketones give no reaction, right? So notice that all of these reactions that we looked at already, aldehydes are always the ones reacting, ketones never react, right? So ketones give no reaction with potassium permanganate. This is because ketones cannot be oxidized unless the conditions are very severe to break carbon to carbon bonds, right? So ketones are really stable in that case, right? So we have this here. We know that potassium permanganate tends to be purplish in color, right? If we're going to have a positive test, we're going to have a brownish yellowish color, right? It decolorizes from purple to a brownish yellow, right? And we generally know that, okay? So the ions exist as manganese 2 plus, which is very pale, right? And then when it gets that manganate type of color, right, it's gonna be brown, right? So we have that there. So it's really the reduction of the manganese. But we've looked at that in redox chemistry in Unit 1, all right? It's just explaining the color change. But we just need to know the color change. So a positive test with the presence of an aldehyde will give it the brownish yellow solution. If it's not an aldehyde and it's a ketone actually, then it will just remain the original color. All right? Any issues here? Notice I ask if there are any issues after every single slide. All right, let me just check to see the chat. All right, so let's move on. Oxidation via potassium dichromate. No, aldehydes will, well, when reflux with excess acidified potassium dichromate, aldehydes are oxidized to carboxylic acids again. So it's the same deal, right? Just different color change, right? Exact same deal for both of them. Ketones don't react, just different color change, right? Now it actually moves from orange to green. That's the only difference, right? And that's because of the change in oxidation states of the chromium 6 plus the chromium 3 plus ions, right? So 6 plus is orange, 3 plus is green, all right? We looked at that before as well. So it's the same process for oxidation with potassium permanganate, just different color changes, right? And we use permanganate and dichromate is because there are really common oxidizing agents, right? They're really common. All right. So let's look at this now. Question: are there any issues with anything here so far? Reactions with 2,4-dinitrophenylhydrazine, reactions with ammonified silver, reactions with Fehling's or Benedict solutions, or reactions with any of these oxidizing agents?
Sir, just to recap, you said with potassium permanganate, it's purple to colorless, and with dichromate, it's
Ooh.
what? Orange to
Okay,
green?
orange to
Like
green. Yeah.
colorless.
For the
Okay.
potassium dichromate, it can be it can be purple to colorless, right? Depending on the concentration of the solvents, right? But like the thing is, usually will actually have this brownish yellow tinge to it. So normally within the labs, you won't actually get a pure colorless solution. So I have this here that if you have a decolorizing, anything decolorizing to brownish yellow, then it will still count as a positive test. All right,
Okay.
all right. So I like making those distinctions, right? Especially for Tollens test, it doesn't have to be a silver mirror. It can be a black precipitate. Go ahead. Think I heard. No, go
So
ahead.
so basically, for example purposes, we just can write to colorless, right?
Yes, you can. Or if you want to like be extra, as some persons may say, colorless or in certain situations brownish yellow. So stuff like that. Um, you can just note stuff. All right. So that's really it. So let's look at the reactions that we have with it, right? Nucleophilic addition. I think this is probably the fastest, the shortest PowerPoint I've ever had. So nucleophilic addition now. This is the reaction that we have here. All right. So the reaction of carbonyl compounds with sodium cyanide, all right, and hydrochloric acid. So the reaction mechanism we're discussing now is a nucleophilic addition of hydrogen cyanide, right? So first we have to create the cyanide, all right? So when sodium cyanide is acidified, this poisonous gas, hydrogen cyanide, is formed. So the production of hydrogen cyanide. And which one of you guys would like to tell me which equipment we should be doing this reaction with? What should we be doing this on though, by the way? Because organic chemistry is a very dangerous ordeal. Anybody can name the apparatus that we'd be using when we're conducting this reaction? Any guesses? All right. Okay, so I'm not hearing a vacuum chamber. Lovely. So a vacuum chamber or a fume hood. Right, so those two things will be fine. And you would probably wear a mask as well to make sure that you still have no chance of really breathing it in. And not like a COVID mask, like a proper gas mask. Like hydrogen cyanide um can kill in extremely small like amounts, right? Like picometer kind of like amounts, like micron kind of like distances. So it's really deadly. So the cyanide ion is the weak acid, right? And hydrogen cyanide acts as a nucleophile in this case. So we have this type of reaction here, but how is this possible? We have to really describe how this is possible, right? This is possible through these reaction mechanisms, right? So the reaction mechanisms for the reaction of hydrogen cyanide with aldehyde, for example, propanol, right? And the ketones, which is propanone. So here we start with um this aldehyde here, right? So which aldehyde is this? May I ask? Can anybody name this aldehyde here? This aldehyde?
Ethanol.
Ethanol. Wonderful, right? So ethanol there. So we are going to have the general reaction that we had from here before. So we know that our cyanide will react with our hydrogen there, right? In that way to create our hydrogen cyanide and then produce our sodium ions, right? In that case. This will dissociate, right, to produce H plus and our cyanide ions, right? So we normally draw it like this because this is the type of bonding that it has, right? So looking at this, right, in this case here now, along this bond here, the carbon oxygen double bond, we know that oxygen is way more electronegative than carbon. Therefore, inductive effect, production of partial charges. Right? Same thing that we see every single day. All right? No problems there. No problems should be there. We're gonna have the partial positive charge, and that will allow for the nucleophile to actually come in there. All right? What will happen there now is that the that one of the double bonds will break, right? So one of the double bonds there will break, or one of the bonds in the double bond, I would say, would break, right? And it will actually leave a oxygen with a negative formal charge, which is kind of unstable there. So oxygen bearing a huge negative charge there, right? But the protons are excess in solution, right, from the dissociation of hydrogen cyanide, right, will actually contribute to the bond and produce an hydroxyl group there, all right? So the H plus from the solvent will be used there, all right? Is everything there fine?
Sir, can you go over it one more time, please?
All right, you want me to draw it or you want me to go over it with this here?
Draw it.
Draw it. All right, give me a second. Let me just draw it out. Hold on. Why is this taking being a little bit difficult? Alright, so let me just get this over with. Want to be using this. Alright, I have to come out of this and then add this in. Alright, so clear screen blank. Alright, so let's just use this. So we're starting with ethanol. Ethanol would have something like this. This would be CH3. All right, so ethanol here. So we know the generally what's going to be happening in the carbon oxygen bond. Oxygen is more electronegative, therefore electrons will actually move closer to the oxygen. Right, same deal there. Right, partially negative charge
Sir, I'm
here.
not seeing anything.
You're not seeing anything.
No.
Can you guys see the screen? Oh, you guys are not seeing the screen. My bad. I don't know why it tends to do that. All right, let me know when you can see the screen again. You can see it now.
Yes, sir.
All right. So polarity in this bond, right? Oxygen is more electronegative than carbon, so we're gonna have a polar bond here. Oxygen is slightly negative, carbon is slightly positive. Right? This bond is weirdly placed. Let me draw it like this. Okay. So we already have hydrogen cyanate actually dissociating to give H plus and the cyanate ions. Let me just put that there. Let me not write it like that. Let me put NC and then that. All right. So we have that there. All right. So what's going to be happening here now? All right. The cyanide ions that we've been seeing or that we have in the solution now are going to be our nucleophiles, right? Nucleophiles are attracted to this positive charge here, right? And this would actually cause this bond to break here, right? What is going to be the result now? This is going to be the result. So let's just look at it like this. Hydrogen, still gonna have our methyl group here. But what we're gonna have here now is a carbon to nitrogen triple bond up here, right? And we're gonna have an oxygen here, but the oxygen now has two extra electrons from that double bond that broke. Because note that the electrons went to the oxygen, all right? Now we know that we also have protons in solution. So the proton that is there in solution will now undergo some amount of attack there, right, from the oxygen. So electrons will now move towards the protons that are free in solution, producing a substance that looks like this, right, with an alcohol group there, right? So hydroxyl. Make sense?
Yes, sir. Thank you.
No problem. All right, so that is what's happening here in our nucleophilic addition reaction. It is not a substitution, all right? It's an addition, all right? So we have that there. So that's what's happening with the aldehyde. The ketone is a little bit different. It's around the same thing to be very honest. But it's the same thing. Okay, I won't. I don't think I would need to explain in depth, right? But what's generally happening here is that the ketone would have this structure now, right? Therefore, our ion will actually have to attack from here, like that, right? To produce something looking like this, R2, R1, and our oxygen up here. And it will actually attract a hydrogen ion, right? And then you know where it goes from there, it just forms the alcohol. So is everybody fine with these two?
Yes, sir.
All right. Let me just check the chat real quick because I actually haven't been looking. Mm, where is it? All right, so I don't see anything that bad. All right, so let's move
It's
on.
okay.
Oh, go ahead.
One question before we move on. Is hydrogen cyanide itself like really unstable, or is there something that breaks it apart in solution?
Well, if it is in solution, then water molecules will solve it, meaning that it will break it apart. Is it unstable though? It's really volatile. Would I say it's unstable? Hmm. It can break apart rather easily. But I wouldn't say it's unstable. Let me actually check real quick before I make any actually positions on that. Alright, because I'm not too sure whether or not it's described as being unstable. Alright, let me just check. Do 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 do. It is unstable. Alright. Thanks for the question. It is unstable. Because I know it degrades easily, so therefore probably it's unstable, right? Um it degrades slowly in the atmosphere though. Alright. So it's non-persistent, it's unstable there. So it would actually break apart easily in order to undergo this type of reaction, right? Cyanide specifically, why it's such an issue, why it's such a deadly poison, stuff like that. You guys did oxidative phosphorylation already in biology unit 2?
Uh, Yes, sir.
So you know your after your Krebs cycle, you create your FADH and your NADHs, right, as electron carriers to your electron transport chain. Your one of your complex, not the cytoquinone, but the last complex, right, on the electron transport chain within your cells, within your mitochondria, right? It helps with the oxidation of the reduction, sorry, of oxygen, right, in order to create water, right? Now, cyanide tends to block the processes of these compounds, right, allowing the buildup of free radicals and other harmful substances within the cells, right? And it tends to do this rather quickly, right? So it is disruptive for respiration in living beings, right? And it anything disruptive to respiration tends to kill quickly, all right. No, this information is used only for chemistry, all right, and only for this type of mechanism, all right. So we won't be discussing this further, but that is why cyanides specifically are particularly dangerous, right? So we must use precaution, well, different precautionary measures, um, and under a fume hood, of course, right, or a vacuum chamber. So we have that there. Reductions now. Lithium aluminum hydrides or lithium tetrahydroaluminate, right? So we have this complex here is often used as a reducing agent in organic reactions. Really used very, very often, right? Aldehydes are reduced by it to produce primary alcohols, right? And ketones are reduced by it to produce secondary alcohols. No, remember your pathways. When we did the reactions of alcohols, we actually oxidized our alcohols, right, in some type of sense, right? We oxidize them to aldehydes and ketones. And now in the reactions of carbonyl compounds, we're talking about the opposite, the reduction to get the aldehydes and ketones back to their primary and secondary alcohols. Right? So you see, if you see on any like type of unit 2 past paper, you see all those products and all these arrows asking you to tell us what reaction A and reaction B and reaction C is, you guys need to know all of these things. Right? So you need to know the backward, the forward, all of these different types of reactions. Right? So we have that now. So the reaction with the lithium hydrodo aluminate, right, is a nucleophile addition, right? The nucleophile is the hydride ion, right, arising from the complex. And the reaction can also be carried out by adding hydrogen in the presence of a platinum catalyst, right? So we can also have that there. And we also saw this platinum catalyst in when we're looking at redox equilibrium in unit 1. Saw it before, right? And it's used wonderfully, right? It works wonders in reduction reactions, right? So we have that there. Any issues? Any questions here?
Can you go over it one more time, please?
What specifically?
Just the whole reaction, like why the lithium aluminum hydride is used.
Okay. So the thing is, no, why it's used is because it's of an excess of hydrogen, right? So it is a nucleophile in a sense, right? Because of the amount of electrons are present within within the complex itself, right? It allows for the formation of hydride ions, right? So it really acts by adding the hydrogen, right, to the compound specifically, right, breaking a bond, right, based off of the complex. It's a type of ligand type of reaction, right? So it breaks the double bond, right, and then based off of the excess hydrogen on the complex, it adds the hydrogen where it can go in order to stabilize the carbocation that would be produced, right? So it's really just about that. So it's a nucleophilic addition in a sense, right? But we don't look at the mechanism. We just know that, hey, this can do that, and what are the products. That's all we need to know for the reductions here.
All right, sir, got it.
All right, problem. So some persons may want to use the platinum catalyst. Some persons may want to use the lithium tetrahydrodo aluminate. Um, we see the LiAlH4 a little bit more frequently. So if you want to use that one, then that's fine. It really doesn't matter. It has to be either two of these reducing agents. All right. So let's have a look at this now. This is really it. So that's really it, for the introduction into these types of reactions. Because what we're supposed to look at, right, is how we find the carbonyl group, how we distinguish aldehydes from ketones using the Tollens test, right? Using the Fehling's or Benedict solutions using potassium permanganate, potassium dichromate, looking at the nucleophilic addition of hydrogen cyanide, and looking at the reductions using the LiAlH4 or the platinum catalyst, right? That is all for this, right? General session here, right? So, just a general recap, carbonyl compounds undergo nucleophilic addition reactions with the cyanide ion. Right, the cyanide ion acts as a nucleophile because it donates a lone pair of electrons to the partial positive charge carbon atom in aldehydes and ketones. Aldehydes are reduced to primary alcohols by lithium aluminum hydride or hydrogen using a platinum catalyst. And ketones are reduced to secondary alcohols by the lithium aluminum hydride or hydrogen using a platinum catalyst, right? So we have that. So we denote that by just having H slash platinum, right? Platinum there. So that is how we write it. So please do not write platinum and hydrogen and platinum. No. Use the correct written form, right? So using that and platinum. All right. So that's it for the reaction for this basic introduction into the reactions of carbonyl compounds. All right.